Hello, in this video, we're going to be looking at the structure of DNA. So let's not waste any of your time. Let's get right to it.
It's hard to breathe, but that's alright. Hush.
Now, when we're talking about the structure of DNA, one thing we want to start with is that this will be somewhat of a review from our material in Macromolecules, Chapter 3, way back when. But there'll be some additional material as well. So just starting with the basics, as we also mentioned in the previous video on one of the conclusions. But if you think about DNA and that structure that it has, you know, when it's in a chromosome state, and again we touched on this in both mitosis and meiosis, it's the idea if you unwind it and lay it flat, as you can see in the diagram, it almost has a ladder type structure where you got your side rails, you've got the rungs along there where you'd put your feet, and those side rails are alternating sugar phosphate, sugar phosphate, sugar phosphates. And of course the rungs are the nucleotides, the AT complementary base pairs and the CG complementary base pairs. And it's that idea of, that's the structure. Some new things that we want to talk about is that we're now going to add the idea of a 3, and that little symbol is red prime, 3' end and a 5' end, and we're going to talk about what that means and why that's important. Now when DNA is replicated, it's made in the 5' to 3' direction. So if you're looking at the diagram on beside me here, at when the DNA is replicated, it's going to be separated from each side. So in other words, the nucleotide bonds will be broken, and so the strand closest to me would be read from the top to the bottom and duplicated. The strand to the far side, on the right side of the diagram, would be read from the bottom up to the top. And that DNA in this diagram example does it best when it's going from the top to the bottom. The other way is kind of like the reverse, so starting at the end of the strand and working back, and that creates a little bit of obstacles, but we're going to talk about when we get to replication in our next video what the cells do, what the DNA does to overcome that. Excuse me. Now, what we're talking about when we say 5' and 3', the sugar, the deoxyribose, is made out of 5 carbons. And if you can imagine a hexagon, you know, a 5-sided figure, if I, you know, got one here, you know, one side, down, across, up, and you know, kind of like a home plate. If you can imagine a home plate, with the point pointing at the picture, the point on the right hand side at the top, that would be number 1, and in the bottom right corner would be number 2, and then go across to number 3, and then up the side to number 4, and then the point which would be pointing at the picture, that would be the 5th carbon. So when you think about those numbers, the 3 carbon end is going to be the back side of the sugar, and the 5' end is going to be like the front, side of it. So that's what it means when it means the end. That means on a 3' end, it's that flat bottom of a home plate that's facing out. And on a 5' end, it's the point of home plate that is pointing out. Well, that's our video over the structure of DNA. When we come back next time, we're going to be talking about DNA replication. So we'll see you then.